Good afternoon, everyone. We're just still waiting for some more people to join us. In the meantime, welcome to the Mind Action Series Life Sciences webinar. My name is Rizal Lowe, and I'm the Sales Manager at Mind Action Series. So our presenters today are Lorraine Keane and Susara Norkia, and they are the co-authors of the Mind Action Series Grade 10 Life Sciences and Grade 8 and 9 Natural Sciences textbooks. Welcome, Lorraine and Susara. Please take note that we are recording the webinar and we'll share it afterwards with everyone. So if you must absolutely leave early, you will not miss anything. We will also share today's PowerPoint presentation with the recording. If you have any questions, please type them in the chat box and we will answer them there. So yeah, let's get right into it. Lorraine, over to you.
Hello, everybody. Yes, I'm Lorraine Keane. Some of you know me and some don't, but I'm very happy to see all the names on the screen. And yeah, I think from the two of us, Susara will be the first person to start and say a few things if we can fall right in with the presentation. Susara, now over to you.
Sorry, can you hear me? I just realized my microphone is off. Can you hear me?
Turn on your microphone.
On now?
But we can't really hear you.
And
there you are.
Can you hear me now?
Yeah.
Okay. Good afternoon, everybody. And number one, thank you all, Copy, for making this possible. Thank you for joining, all of you. I know it's very busy when you're a teacher. And if you can have 1 hour free, it's absolutely great. So we appreciate it that you share your time with us. We try to put this presentation together on the concept that was written in the ATPs, just to have something to work from. And then the next thing I would like to tell you is that we discussed what is important for a teacher to know, to share with the children exactly what will be asked in the exam. I wish I could tell you the questions, but the point is, how would it be marked? What is important? And help your learners in such a way. Some of you are teachers that's been there for many years, so you don't need us to tell you about the subject and the core principles, but I know it's important to have a bit more insight in how things will be marked. Some of you, maybe it's just a short time in teaching, and if you would like to make sure of definitions, please feel free to use the chat room. I just want to ask you to not not to talk, but just to write it in the chat room. I will definitely answer that, or I will ask Lorraine to give you an answer. And if you feel better or it's easier, do it in Afrikaans. En ons sal jou in Afrikaans antwoord. So this would be, the presentation will be in English, but if you in any time want to ask something in Afrikaans and you want us to explain, please feel free to do that. A personal thing from my side is just that it's absolutely great to sit with all of you on a platform, if I can say it that way, it sounds terrible, and to share time with other people that are that love the same subject. This is an absolutely wondrous subject to be able to teach our learners. This is absolutely fantastic. So I'm giving it over to Lorraine. The reason for that is she is the chief marker. I think you know that by now, but she is the chief marker of this paper where this part of the subject is questioned. So Lorraine, I'm going to have a drum roll, then it's yours. Lorraine, sorry, we can't hear you.
Here we are. Okay, let's get started again. Thank you, Susara. Susara is the CEO of Amazing Brains, a company that provides teaching, training, and for teachers and caregivers at early childhood development centers. They're doing wonderful work, but she used to be a life sciences teacher, so she also knows what she's talking about. I'm going to open the presentation now, so give me 2 seconds. I've practiced this, and there we go. And it's slideshow, and there we are. With our topic, Introduction to Genetics, I just have to say that it really is an introduction. We didn't go into topics such as cloning and GMO products, because we can do that at a later stage. We can lift the standard of today's introduction to genetics. But our feeling was that with the name of the topic, we really want those teachers who might teach genetics for the first time, or those who've been teaching it for several years but need some material. We want you to feel comfortable. And as Susara said, please put your questions in the chat. Okay, so we looked at the CAPS document, and Susara put together what we need to teach our learners and where we touch on genetics. And the first thing, of course, in grade 10, if we just look at the FET band, is the cell structure and the chromatin network in the nucleus. And then, of course, to a certain extent, also mitosis, where we have chromosomes. Then grade 11, biodiversity. And in grade 12, the concepts of inheritance, types of dominance, genotypes, phenotypes, dihybrid crossings, etc., etc. And this is what we will touch upon today. Okay, concepts of inheritance. If you go and look at your ATP for this topic, there's a long list. Can I just say that I find in my school that I don't tell the authorities, but that I do human reproduction and animal reproductive strategies after genetics, simply because the genetics, the topic connects better for me to meiosis and DNA and RNA. Then we do reproduction because together the previous 3 topics in reproduction will then lead into evolution, which I also prefer to do first. Jean and I have spoken about it, but we know what the ATP and the CAPS documents say. So some of the concepts, if you look at your grade 12 textbook and all the documents, is that you must revise the structure of a nucleus. And from the chromatin network in grade 10, we go over to the DNA in grade 12. So yes, it is important that they know the structure of the nucleus and that they have an idea what all the structures are, what their functions are. Now, just before cell division, after replication, whether it's meiosis or mitosis, the chromatin network becomes visible as chromosomes. Isn't this a fantastic photograph? Now, what we have here are replicated chromosomes, which means that after DNA replication, so you will find that these double strands are now after replication. And our candidates have a problem with that. I will say something more about that later. There we have chromatid, and then the whole structure, a chromosome, and the 2 chromatids bound by a centromere. After our memo discussion in Pretoria at the end of last year with a view to the marking of the NEC in November last year, it was decided, and it will appear, the diagnostic report is out, that from now on the only terms to be used will be chromosome and chromatid. Chromosome as the whole structure is here, chromatid for 1 strand. Okay, So let's get back to the nucleus. The chromatin network is broken up into chromosomes. Chromosomes consist of DNA wound around the histones, and it contains several sections that will code for characteristics. And we know a gene is a section of DNA. There you've got a gene and its section of DNA. Deoxyribose nucleic acid. Now, a homologous chromosome pair is also one of the terms that must be known. It's two chromosomes with a similar shape and size, not identical, but similar. And why not identical? Because although they carry the same genes, the alleles of these genes might differ. We'll get to that. And here we have it. A haploid cell has a single set of chromosomes, and a diploid cell, a double set of chromosomes. Here you've got your single set. This is what we will have in a gamete, and this is what we will have after fertilization, a diploid cell. Now you can see chromosome, this is a gamete. Now, when we look at this, we will have a paternal chromosome and a maternal chromosome. For us, with our 46 chromosomes, 23 from the father, 23 from the mother. Same here, paternal, maternal. Here you've got it, a paternal chromosome, a maternal chromosome. And homologous pairs of chromosomes after DNA replication. There you will have the full chromosome replicated chromosomes, and they are a homologous pair. Similar structure, same genes, but maybe with different alleles. It will depend on the genotype, the genetic combination. Here we can see there is a gene coding for a characteristic. How are these genes expressed? Paternal, small t, and a maternal, small t, probably. Okay, genotype, small t, which means that this one is perfectly homozygous for those characteristics. And then we get another homologous pair. Here we can see paternal, maternal, but the paternal chromosome, the genes, the alleles are dominant characteristics presented by capital letters, while the maternal chromosome have recessive characteristics represented by small letters. And it's both big T, because it's the DNA and its replication. DNA and its replication. So there we've got it. And the genotype will be big T, small t. Heterozygous for this specific characteristic. Now, during meiosis, prophase 1, and that is something that's already been done with your grade 12s, we have crossing over between the two chromosomes of a homologous pair. Afterwards, we have chromosomes with exchanged genetic material. There's an exchange of genetic material between homologous chromosomes. And that is important for genetic variation. Please note that a chiasma is the place where two chromatids touch. In some textbooks, they talk about chiasma formation or chiasma forming. That is not the process. The process is crossing over, and that is the only term to be used. There's an exchange of genetic material contributing to genetic variation. Genetic variation, and that's a topic for another day because our next workshop will be on evolution. But apart from genetic variation in the offspring, variation also leads to natural selection. As I've said, a story for another day. The inner chromatids will swap genetic material, so there you will have variation. Okay, next concepts will be Mendel and his so-called laws, not proper scientific laws, but they are known as laws, and the concepts of phenotype and genotype. So, Sarah, is there anybody with a question, or is it too early days yet?
I think you're doing a good job with the
Okay, not yet.
introduction. Yes,
Right,
this
that's
is
fine.
this work that you've done up till now would be the work that that has been done with a matrix or even grade 10s for that
Okay,
matter.
yeah. So they should know this. So thank you then. Mendel's law, the phenotype and the genotype. Mendel was an Austrian monk who lived in the middle to late, well, early to late 19th century. It was a little bit younger than Charles Darwin, and just think what could have happened if the two of them had the chance to meet. You know, in those days they didn't have WhatsApp or email or anything, and Mendel published his work in a fairly obscure German paper, so nobody else really knew about his work. But we still have Mendelian principles that we use. Now, Mendel was fortunate in his choice of garden peas for his experiment because the garden peas had contrasting characteristics, purple and white flowers. And this is a classic example, and I think you all know about Mendel and his garden peas. His work was not recognized until the early 20th century, and then it was specifically three scientists, and our matrics don't have to know that, but it's important that they know that there were men who reworked Mendel's notes and books that he had, and looked at the Mendelian factors that he talked about and then reworked them to what we know today as genetics, of which father is regarded, Mendel is regarded as the father. And there you can see the little garden, of course, a re-garden of Mendel's experiments with his garden piece at the monastery where he worked. Okay, Mendel's work led to three laws. Now, Mendel didn't use the terminology that we use today. So he talked about factors and the one coming out and expressing itself. But let's look at the law of dominance, his first law. Mendel said that the law of dominance means that in the heterozygous condition, the dominant characteristic will be expressed in the phenotype, and the recessive characteristic will only be expressed in the phenotype in the homozygous condition. So what do we say? Here we have a tall pea plant and a short pea plant. Tall is dominant over short. In the pea generation, both the alleles for the length of the plant were dominant tall. And in the plant that it was crossed with, both the alleles said recessive short. Now, when you make a crossing and you get in your F1 generation, each of those individuals will contain one dominant allele and one recessive allele, and therefore the dominant characteristic will be expressed in the phenotype. So heterozygous means one dominant, one recessive allele for that specific characteristic. If we should cross two of these heterozygous individuals, we will get a phenotypical ratio of 3 tall plants to 1 short plant, or 75% of the F2 crop will be tall and 25% will be short. And we will look at the crossing soon to find out. But the point is, the short plant, we can know without any doubt that it is homozygous for the recessive characteristic. Good, let's continue. The law of segregation means that the alleles of a gene remain separate and they do not contaminate each other in the F1 or the hybrid. This is more the Mendelian way. Today we can say the two alleles for each trait separate during gamete formation. They are not linked. They don't go together. Whether it's homozygous or heterozygous, when gametes are formed, they separate. And that is the law of segregation. Just a tip, especially for those matrics who take history as a subject. They often read words instead of the sentences of a question. Just note that the segregation does not mean the political term that we had during apartheid, and pardon me for mentioning it, but when we mark matric papers, every now and then we find that whole centers will write about segregation in politics. This segregation means separation of alleles during gamete formation. And in the last of Mendel's three laws, the law of independent assortment, the alleles for the different traits or characteristics are distributed to sex cells independently of one another, which means that the black hair and the brown eyes do not have to go together. And the blonde hair and the blue eyes do not have to go together. This boy is a mix, his hair, of the parents. There might be more alleles determining hair color. We know there are. Blue eyes does not have to go with the lighter shade of hair. So just keep that in mind. For our matric learners, they have to know the three laws of Mendel. They have to be able to state the laws. When we look at crossings and types of dominance, before we start with dominance, just monohybrid crossing. Monohybrid crossings refer to the inheritance and crossing of one characteristic, either fur color, either hair color, etc., etc. So they will have genes, and with the alleles, in this case, the gene will be a fur color, and the alleles will code for either black or gray. Pretty rabbits, isn't it? Black is a dominant characteristic, and gray is recessive. It doesn't always have to be. In some cases, white will be dominant, and gray or black or brown will be recessive. Now, we look at the crossing and what the possibilities for offspring are by using a specific template for our crossing. Here we have a monohybrid crossing. We're going to look at two rabbits, a black rabbit, which is homozygous for the dominant black characteristic, and a gray rabbit, which is homozygous for the recessive gray fur color. Just a side note, it is simply a convention to write the males on the left and the females on the right. You will often find in some textbook or in exam practice books, that it's swapped around, female on the left, male on the right. The only time when you have to write male or female with the phenotype is when it is a sex-linked characteristic, to which we will get later. This is homozygous individual. You do not write black homozygous for phenotype, because the term homozygous refers to the genotype. Phenotype, physical characteristic. In Afrikaans, phenotype voorkoms. Genotype, genetic composition. So let's have a look. This is our first parent generation, and there we have the two alleles. Meiosis occur, and then we have the gametes. Now, you and I all know that in males with the formation of sperm, we will know that there will be 4 sperm cells formed from each parent cell. And in the formation of an ovum, the same thing will happen, although 3 of those cells will form polar bodies and only 1 will develop into the ovum, sorry. I'm not having omens here. But here we work with statistics, and 2 gametes will be the smallest number we can use to work with our statistics. Now we have meiosis gametes and then fertilization occur. You will often find that fertilization is done like this, with lines. And we have to look at all the possibilities. Will this gamete fuse with that of the female or with the other one? Or will this male gamete fuse with this female gamete or the second one? But this is our F1 generation that we will get, and we indicate as such. We propose that candidate students rather use a Punnett diagram. And the reason for that is simply it helps the student to organize the alleles of the gametes so much easier and to see so much easier what is happening. And they will get a mark for the gametes. They can get a mark each or just one. They will get a mark or just one for the offspring in the Punnett diagram. Right, so we said it's much better, but you have to use the template. Now, at the end of the crossing, it will be the F1 generation, the first filial generation. And we need to record them, to write them down by their genotype as well as their phenotype. Now, in this case, if we look there, we had 100% capital B, small b, 100% heterozygous, and 100% in the phenotype that will be black, as we can see here. So we need to write the genotype. This is the genotype, the phenotype, that is what it is. And in this case, it was 100%. So this is included into the presentation that you will get. And by the way, if I haven't said it, this presentation will be provided to you in Afrikaans as well. So die Afrikaanse mense kry 'n Afrikaanse PowerPoint. Ek werk aan materiaal saam met Susara om vir julle ook voorbeeldvrae oor genetika met memorandums te gee. I'm working on material to give you questions, example questions, as well as the memoranda. So hold on. Let me just set my matric test for next week and then I'll get to you. Oh, well, I have started. But this is the template that must be used. Now, often in memoranda, you will find that at the bottom of the memo, they will write P1 plus F1, 1 mark. Meiosis fertilization, 1 mark. And then the candidates write it there as well. This is how they get the marks. Position of the P1, the F1, and a tick. Position of meiosis and fertilization, and a tick. Then they get a mark for the correct phenotype and genotype of the P1 as well as the F1 generation. They can get their gamete mark here, or they can get it here. And they can get their genotype mark here at the bottom or be in the Punnett diagram. Are you still with me? Okay. Now let's take the offspring of the rabbits and cross them and see what will happen. These were we got a totally black furred rabbit, and let's take 2 black furred rabbits and cross them. They are the gametes. So the gametes of the rabbits will either be capital B or small b. There you have it in the Punnett diagram. And look here. In the offspring, we will have a capital B homozygous for the dominant characteristic, 2 heterozygous individuals, and 1 homozygous for the recessive characteristic. Now, if we have to write this down, we say the genotypic ratio of the offspring is capital B, heterozygous, homozygous, 1 to 2 to 1. But because both these genotypes will be black, the phenotypic ratio will be different. 3:1. And let's look at how the ratio is written. This is the acceptable way for which our learners will get marks. They must also look at the question. Does the examiner ask, give the genotypic and phenotypic ratios? Or does he ask, what is the percentage chance? If he had asked, what is the percentage chance for two black heterozygous rabbits to get a gray baby rabbit, then you shouldn't have set the ratio. They should have worked it out and set a 25% chance. Right. Now, we've just had an example of complete dominance, where the recessive characteristic will only be visible if the individual is homozygous for that recessive characteristic. So complete with the black and the grey-furred rabbit, well, there we have it. Okay, and this is also what Mendel said, because Mendel had contrasting characteristics. Then we go further. The second type of dominance is incomplete dominance. Ladies and gentlemen, please note, colleagues, that there is no matter of dominance. We either say incomplete dominance or the absence of dominance, but this is the preferred term of the examiners. No characteristic is dominant over the other. So it is incomplete dominance. And the offspring exhibits an sort of an in-between characteristic. Snapdragons is a classic example. If you cross red snapdragons, Leobekkies, with white snapdragons, the offspring will be pink. Because we don't have dominant and recessive, we use capital letters for both conditions, and both capital letters its own. So here we can say red, it will be capital R, capital R. White, capital W, capital W. So homozygous, and then the offspring with an in-between characteristic, with a mix, will be heterozygous, but for two capital letters. I've read an article where they argue, shouldn't we use small letters because there is no dominance? And then they went back into, yeah, but we used to small letters being for recessive as opposed to dominant. However, this is what we use. Isn't this a pretty picture? And here we have it. If we breed the F1 heterozygous in-between condition, if we interbreed them, this is what we get in the F2 generation. The two contrasting characteristics, and then 50% will be once again the in-between characteristics. So phenotype will be white to pink to red, 1 to 2 to 1, or 25%, 50%, 25%. And the genotype will be WW, RW, RR. I actually didn't know it, but some carnation, natural occurring carnation colors also show incomplete dominance. This third type of dominance that we're looking at is codominance. Now, in codominance, both characteristics will come out. So the offspring will show both the characteristics. This is a type of camellia, and if you look at the flower, it was a plant with white flowers crossed with a plant with red flowers. And the offspring will be red and white. Not pink, red and white. You know, roan cattle or even dappled horses that will be a mix of both. Here you had, I don't know which was the cow and which was the bull, but you had a white, let's say a white bull and a red cow, and the calves will be roan, not white or red. And you find that in plants as well as in animals. Codominance. Remember codominance as opposed to incomplete dominance. Yet another flower type, impatience this time. Right. Now, I like this summary. Complete dominance, incomplete dominance with an in-between, and codominance. Look there. Both characteristics visible. I actually thought it's so simple, but it's so effective to use this. Right. Let's look at a few more of the topics. Now, we've talked about codominance now. So we're going to look at blood groups and codominance. Now, two alleles in heterozygous individuals, for example, blood types, will show us an interesting case. When we look at the ABO blood group types, we have 4 blood types. Type A can either be homozygous with 2 dominant alleles, or it can be heterozygous with 1 dominant. Oops, I see there's an A that turned into a small a when I did this, a small a. Sorry, that must be, and I'm going to change it immediately because this is one of those that jumped into its own. I hope I don't get any more. Are we back? Can you all see me there? Complete dominance in type A. We are used to say that blood groups is an example of codominance, but we must remember it's only one of the blood groups. A and B show complete dominance, right? So they're either homozygous or heterozygous. Type AB shows codominance. And I'm going to show you now in a moment why it is like that. And then type O is homozygous for the recessive characteristic. There is no dominance again. So our learners must know the way that the alleles are written, and this, a capital I with a superscript A or B, or the small I for the recessive characteristic. These are the only ways to write it. No other way, please. We do find different ways, different textbook, different articles that we read, but this is, according to previous diagnostic reports, the only way. And you have, if you haven't received a diagnostic report yet, I have our life sciences diagnostic report. It was sent to schools. I've got the whole booklet for all subjects. Please just let all copy know, and they will get us to send it to you, because you've got their mail, they've got yours, and we will let you have it. That is now for the diagnostic reports of the past few years and last year's. Okay, so now I think you are okay, you understand. We have complete dominance with A and B. Type O is a recessive homozygous condition, and only blood type AB is codominance. And here we can see it's about the antigens that are proteins found on the red blood cells. There you can see them, and then the antibodies produced by it. AB, the body does not produce antibodies against blood group AB because it's got both types of antigens. O has no antigens, but both antibodies A and B are produced. If we look at this diagram, we can see why AB can be or is the universal acceptor. In a pinch, a person with blood group AB can receive and survive from blood type A, blood type B, and blood type O. Blood type O, on the other hand, is the universal donor. Universal receiver AB, universal donor O. The anti-A and anti-B antibodies is fine with blood group A, blood group B, blood group AB. But do not give an O person any of the others. So O, universal donor, AB, universal acceptor. I didn't put anything about the rhesus factor in here. Just maybe if you can carefully listen, the rhesus factor is also an inherited factor, and where they say you are blood group A positive or B negative. The positive is dominant, the negative recessive. Now, two dominant parents will make a dominant baby. Two negative parents, no, we're not doing maths. Two negative parents will make a negative baby. And a positive parent with a negative parent will make a positive baby. And it's fine if both mom and baby are positive and the father is negative. But if the father is the positive one and the mother negative, the baby will be positive, and that is when we get a rhesus baby. And I always tell my matrix that is one of the reasons why pregnancy should be planned, because then the doctor will know your blood group and that of your husband, and they will know that they must take care that if you get pregnant, there might be a problem with the blood groups. That's just a side comment. Okay. I don't know, Days of Our Lives. Who watch Days of Our Lives, you don't have to put up hands, and with kids. There were two characters, the characters named Hope and Lexi, and both had a son. And as these series go, these babies were deliberately switched in the hospital. Now, we know a little bit more. So we can see, if we look at the blood types of Hope and her husband, Lexi and her husband, and the babies, we can tell them which baby is which couples. Not like my matrix who said, yeah, but Hope's husband had an affair with Lexi and vice versa. Please tell your kids they must take it that we still live in a good world where everybody is with their own people. But there you've got it. Baby type O. And that is really where you start. Type O and type AB. O and AB are the two types where you know exactly what the genotypes are. Hope's husband is AB. So, for starters, he cannot have a child with a blood group O, because O is small I, and if he is IA, IB, where does a small I come from? Hope could be IB small I, but she could have, so she's B, but she could have the type A child if her small I comes together with her husband's A. Lexi, on the other hand, are both type B. So why do we say their child is O? Because if both of them are heterozygous, they can each give a small I to a child, and there the baby is small I, small I. Here's Hope and her husband. So their phenotypes will be AB, A, and B. No possibility for a child, O. By the way, here we can see, and in the next slide that I'm going to show you, why blood groups cannot be the only way to establish paternity. By now you have done sorry, where am I now? By now you have done DNA profiling. Sorry, I wasn't there for a moment. So you know that DNA profiles are actually an excellent way to establish paternity, even maternity, even any other genetic relationship. This is Hope's husband. There are many millions of men, IA, IB. So even though Hope is the mother, what her husband, or if this was a boyfriend of a girl, could say, I'm not the only IAIB, I'm not the only AB blood group person in the world. Then we use genetic profiles to totally establish paternity. Okay, then Lexi and her husband, IBI, IBI, and there you've got them. And for them, it is possible to have a baby, blood group O, a 25% chance. Remember, once again, learners could be asked to work out the blood group of the possible children, or they could be asked what is the ratio, or they could be asked what is the percentage chance for these two parents to have a baby with blood group O. And if they work towards blood group O, don't even go to homozygous IBIB or IBIB or IAIA. Because if the baby is blood group O, there must be two recessive alleles, one from each parent. Okay. Blood group is also an example of multiple alleles. We have 3 alleles, IA, IB, and I. 2 at a time code for a blood group, but there are 3 alleles. We have other characteristics as well. Skin color, for instance, if I'm not mistaken, we have 5 pairs of alleles, 5 genes, 10 alleles in total that will code for skin color. So it's multiple alleles. Well, yes and no. Right, then the inheritance of sex. And I wonder, Sheila and Roselle, Susara, before we go into sex and sex-linked diseases, if we shouldn't have a 5-minute comfort break, what do you say?
Hi, Lorraine. Yes, 100%.
Yeah. So
No problem. You can take a break.
Yeah, let's say so
agree.
5 minutes just before 4 we'll start again.
Okay, 100%.
Good.
So, yeah,
Okay, thank you.
participants or attendees rather, you can also just take a quick break, have a quick coffee, and then within 5 minutes we'll reconvene.
Good. Excellent. Thank you. Are we ready to continue? Give a minute or two more.
Yes, Lorraine, I think we can continue.
Okay. Right. Ready,
Alright.
Susora?
Lorraine, before you start, can you hear me?
Yes.
There's been a question and I'm going to read it to you. Please confirm. NSC includes rhesus factor? Question. Or is that IEB only or just for interest?
Right. It is not for NSC. I'm not sure about the IEB. I went through the curriculum some time ago, but I'm not sure. But you guys will know, but it's not for NSC. For NSC, it's just for interests. Sad, I know, but alas, it's not there. But I feel our learners should know it because it's something to go out into the world with. But it's not part of the examination guidelines. And by the way, while I'm talking about examination guidelines, once again, the NSC exams have an examination guidelines document for most subjects. If you do not have that, please also send a message to All Copy and I will also provide you with that. It's available in Afrikaans as well as English. It was the last dated 2023. It has not changed. The ATPs came out for NSC sometime earlier this year. That's another document that you should have access to.
Lorraine, it's just been
Yes.
confirmed. Colette Nadel now, she's the educator at Curo Durbanville, said
Yes.
it's also not for IEB.
Okay,
Just
so
thank you
rhesus
so much for
factor,
that.
not NSC, not IEB, for both, just for interest's sake. Okay, thank you so much. But as I say, it's something that I think our learners should know. You know, we are under huge pressure to perform well in exams, whether you teach IEB or NSC, whatever. The pressure is there. And when the kids do well, they applaud it. And when they don't, people look at us. But I honestly think that we should send our matrics out in the world. It's the last chance that we have, with enough extra knowledge to make their lives easier, to color it in, to broaden their horizons. Something such as rhesus factor. They also don't have to know about the antibodies and the antigens on red blood cells, but let them know why there is something such as universal donors and universal acceptors. And by the way, if you are in dire need of a blood transfusion, a chimp with the same blood group can help you. We share our blood group typing with chimpanzees. They are, after all, our closest family in the animal world, but let's not go there. Okay, we've had a break. We can continue. Inheritance of sex, male and female. Once again, I might be wrong. My idea of sex, male and female, is what we have here in the chromosomes. Now, we also know that genetically, we have, in between us, we have different combinations of chromosomes and expression of male and femaleness. Gender is, as far as I'm concerned, something else. Is how a person experiences his or her sex, whether it's transgender, queer, lesbian, homosexual. That, as far as I'm concerned, refers to gender. However, this is my personal opinion, you will find, and that is important that you tell your learners, that in question papers, they will refer to gender, meaning male and female, as we have in these two karyograms, also accepted to call them karyotypes. So the gender on the left will be male, the gender on the right will be female. So the term gender and not sex will be used. Our textbooks still talk about inheritance of sex or sex determination, but yes. Here we have, as I've said, karyograms, and we know that we have 46 chromosomes, of which 23 pairs, of which 22 pairs are autosomes, and the 23rd pair are the gonosomes determining male or female. If the two chromosomes are different, X and Y, it is a male. Now, these chromosomes are single, they look like single chromatids at the moment. Now, if we look carefully, we can see the chromatids are just lie very, very close to each other. So we can also get them where they form chromosomes with two chromatids, where you will have 4 for a pair, for instance, where it's a proper X and a Y. If the gonosomes have the same size, or it's indicated as XX, then it is female. Any genetic disease or condition that lies on the X chromosome is sex-linked. The Y chromosome is simply too small. There's a few characteristics that is coded for by the Y chromosome, but genetic conditions, sex-linked, will lie on the X chromosome. They have asked before, what is an autosomal characteristic or autosomal genetic disease? That is a chromosome or a disease that lies on one of the autosomes. In other words, one of pairs 1 to 22. Right. Here you can also see what we mean by homologous pairs. Same size, similar shape. Okay, X and Y are the gonosomes for females and males respectively. Male inheritance, female inheritance, and there's a 50-50% chance that a baby will be female or male, that the genotype will be XX or XY. Now, our learners must know this, and they must be able to show with a genetic crossing. According to this template, for which they can get 6 marks, they must be able to show how sex is inherited or how the determination of male and female occurs. Once again, for interest's sake, during meiosis, when we did that, we talked about abnormal meiosis and we talked about Down syndrome. We also know that we often, not quite that often, but yes, more often than we would like to, we have non-disjunction of chromosome pair 18 leading to Edwards syndrome. We've got a 13 non-disjunction problem that is sometimes found, and so we can continue. These are autosomal problems. What about a missing X chromosome, only one X? Because the other gamete had the X at XX and the one gamete no X. So when you only have one X chromosome, it is Turner syndrome. XXY, Klinefelters. It's not for the exams, but tell them about it, please. Even triple X, even XYY. So please tell them, because once again, our learners need to know what's out in the world. Okay. So now let's look at a few genetic problems. Autosomal recessive albinism. It's a genetic condition inherited from both parents. It is recessive. and it's on autosomes. No pigment with the resultant problems. Melanin is needed for normal receptors in the eyes and ears to form. And we get people, we get animals that are albinos without any pigment. Now, albinism occurs amongst all people. It is just more visible amongst people who generally have darker skins, but it's not restricted to any specific group. We also have an
Lorraine, we can't hear you. Please put on your microphone.
Okay, can you hear me now?
There you are. Thank you. I think you need to start from before the lines.
From where didn't you hear me?
From the lions. You were talking about the
Okay, I
animals,
was
and then
talking
it goes
about the
before
lions.
Yeah.
Okay,
Yeah.
I'm not going to answer. Susara? There you are. Okay, sorry about, I don't know what happened because I was just sitting. But anyway, here I'm back again. Okay, so we've got the autosomal recessive condition, and I've said that it occurs amongst all groupings. It's not restricted, and we need to tell our learners about it because we know that in some communities it's still regarded as something that's not to be tolerated. There's a lot of intolerance, but it's simply a genetic recessive allele where a homozygous recessive individual produces no pigment. And we get it with animals, people. But then we sometimes get that animals are white. I know you know the story in the video that was made, or the film, The White Lions of Timbavati. There was a book where the lions really are white. They're not that golden brownish color, but they're not albinos. If the hair is white, if you look at this lion, it's got dark pigment on the nose and around the lips, and the eyes are dark and it's rimmed with dark eyelashes and eye sockets, or the rim of the eye is dark. So don't get confused with white animals and albino organisms. Sickle cell anemia. Now, the matrix must know about albinism. So when they get a question about albinism, they are supposed to know. They are supposed to know that it is caused by a homozygous recessive condition. Sickle cell anemia, they don't have to know. If they get a question on sickle cell anemia, the information will be given. Sickle cell anemia, we know that it's the most common genetic disease amongst African American people. It's also, and that is because of slavery of the past. And we know that sickle cell anemia is endemic to Central Africa. It's also the area where we find malaria. And the benefit of having sickle cell anemia is that you can't get sick from, you are immune to malaria. So a homozygous sickle cell person could die and they don't survive long. A heterozygous person will have some normal blood cells and they will survive and they are immune to malaria. The reason why it's painful is because these cells cannot fold like other normal red blood cells do when they move through tiny capillary vessels. Now, this is an example of co-evolution. What was first, we don't know. Research is going on, but yes, interesting. Hemophilia is a sex-linked disease. I've referred to it earlier. It is carried on the X chromosome, and sufferers lack a blood clotting factor, and injuries cause excessive bleeding. This is not a bad case of hemophiliac bleeding. It's really I had a cousin with hemophiliac bleeding. It was like sacks of blood hanging on the joints. But the point, our learners must know hemophilia. And they must know that it's caused by a recessive allele on the X chromosome. Men only have one X. So if they inherit the X chromosome with the recessive allele from their mothers, they will have hemophilia. Women have two X chromosomes. And if one is for the hemophilia, the recessive allele, they still have the normal dominant allele that can mask that recessive allele. Women will only suffer from hemophilia if both alleles are recessive alleles. In other words, it's if a woman is X small h. Now we know there are different types of hemophilia. Our learners learn about factor 8, a lack of factor 8 blood clotting protein. And this is just for interest's sake. They don't have to know that. And it's also a fact that an embryo that is homozygous, a female embryo, homozygous for the recessive allele, will result in a miscarriage very early on. Now we've had where the examiner asked a mother who is a sufferer, small h. Please tell your candidates not to write the answer as this is not possible and not do the cross. They must take the question at face value and use the information. Okay, so here we have a male without hemophilia and a woman without hemophilia. We cannot say she's a carrier. I put it in brackets. We don't know from the outside whether she has this allele or not. So as far as her phenotype is concerned, she does not have hemophilia. We have to say male and woman and female, or man and woman. I put both in here because it's a sex-linked disease. Then we have the genotypes, the gametes, and here we have the crossing. And if you look at below the Punnett diagram, we have 4 different genotypes. X, a capital H, capital H. That is a woman without hemophilia. Then X capital H, X small h, it's a woman without hemophilia, and that is what the candidates must say. But depending on whether it's a paragraph question, they can say she's a carrier, but that is not her phenotype. Then we have a male, a man, who is X capital HY. He does not have hemophilia. And don't say he's normal, because what is normal? We say he is a man without hemophilia. And then the last one is X small a, Y, a man with hemophilia. So the ratio to female with hemophilia, male without hemophilia, and male with hemophilia is 2 to 1 to 1. What if they ask you what is the percentage chance for a child to be born with hemophilia? It's 1 out of 4, it's 25%. But whoa, wait. What if they ask you, what is the chance for a boy to be born with hemophilia? 2 out of 4 are boys. And of the 2 possibilities for boys, 1 will have hemophilia. So there's a 50% chance for a boy with hemophilia. Got that? Now, hemophilia actually changed the history of the world. Queen Victoria she didn't have hemophilia, but she was what we would like to call a carrier. And they think it was caused by a mutation in her father because he wasn't a hemophiliac. But if we look at the history, she probably inherited from him. Albert was healthy, no problem. Alice, her daughter, inherited the recessive allele. Alexandra, Alice's daughter, and the Tsarina that was married to Tsar Nicholas II of Russia, she carried the recessive allele. And her recessive allele matched up with the Y of Nicholas, and they had 4 daughters, Olga, Tatiana, Maria, and Anastasia. And their 5th child was the long-awaited Tsarevich, Alexei, or Alexis, and he had hemophilia. And it was their desperate need to get help for Alexis from, amongst others, Rasputin, that was part of what contributed to the Russian Revolution. Not a total cause, but it contributed.
Fine, there's a question.
Yes.
Go back to the previous slide. The question, the previous one, there you go. The question is, may learners write the word is instead of using an equal sign?
Mathematically, the equal sign is correct.
Okay.
But I don't think they will be penalized for that. But mathematically, so. They said that is the way to write the
well, let's say life
ratio.
Science is a science.
Yeah.
And
yeah.
linked to mathematics, so we prefer the equal sign.
We prefer the equal sign. And one of these meetings, we were actually told that we must teach our candidates to use the correct way of writing the ratio. And this is the...
If the candidate's in front of you, I mean, the paper, and you're marking, sure, there's nothing to learn from it, so you will get, or she will get the mark.
Personally, I will give them mark because we will mark positively and the rest of the ratio will be correct, but the way to teach the learners will be the equal sign.
Thanks.
Okay. So please ask your questions, people. It is important, and this a seemingly simple question might be something that everybody wonders about. So please ask. Okay. It is interesting that the current royal family also, Queen Victoria is also their sort of ancestral. Victoria was called the mother-in-law of Europe because her and Albert's children married into all of the royal houses. But the Crown Prince Bertie, who became king after Queen Victoria, was not hemophiliac, and neither his wife, so she didn't carry it. So the current royal family does not have the allele for hemophilia. The other sex-linked disease or condition that the learners must know is color blindness. And we get different types of color blindness, and the red-green color confusion is an X chromosome. It's also called Dalton, because James Dalton, scientist, previous century, had color blindness, and he worked it out and described it. Green and red light receptors, because our receptors can pick up red, green, and blue light. The red and green receptors on the X chromosome, the blue receptor is on an autosome. And if you can read the numbers and the figures on these 4 circles, you're okay. If you can't, there could be a problem. Don't despair, go for an eye test. Okay, they must know. And usually the X is indicated with a capital D or a small d. Something else to tell your learners, now in November, we had many learners who knew the story about a sex-linked disease, but they wrote the H instead of the D, and the allele was, or the way they had to write it was given. And another tip for our learners, tell them to read a question from the top. Don't start with the actual questions, read the introductory paragraphs, because usually there is a lot of information for all answers that they must know and that they can use. Okay, then we get to pedigree diagrams. Now, pedigree diagrams are important because they always ask some sort of pedigree diagram. Now, here we have 3 generations, 1, 2, 3. A generation isn't a married couple, another married couple, another married couple. It is the lines, grandparents, parents, children. Squares represent males. Circles represent females. Squares or circles that are shaded or colored in or black represent affected individuals. White or not colored or unshaded represent unaffected individuals. Now, you can't just say affected. It will depend what the question says. This could be a question about colorblindness. Or it could be a question about the inheritance of long noses or the inheritance of rolling your tongues. So it doesn't need to be affected. You will look at the key that is given or the information in the introductory paragraph. By the way, in most of the cases, the key is given. But candidates should know squares are males, circles females, shaded affected, unshaded not affected. Okay. Then we continue to dihybrid crosses. We're doing good with our time. There's time for questions. Please put your question in the chat. With monohybrid crosses, we have one characteristic involved. With dihybrid crosses, two characteristics involved. Alleles for different traits are distributed to sex cells independently of one another. Remember Mendel's law of independent assortment? And this is illustrated by dihybrid crossing. If we look at this example, this is now once again back to the pea, seed shape, and seed color. Round seeds is a dominant characteristic, wrinkled seeds recessive. Yellow seeds dominant, wrinkled seeds, green seeds recessive. Here we have heterozygous round yellow seeds. Heterozygous round yellow seeds. And look at the possible gamete combination. We sometimes find that they write RR and YY. So the one gamete will not code for seed shape and the other one not for seed color. And they have difficulty understanding that. The two letters must be together. They can't write RY, RY. No spaces in between, no commas in between. So when we look at the first individuals, capital R, small r, capital Y, small y, what are the possible gamete combinations? The capital R, remember, according to Mendel's law of independent segregation, could have paired up with a capital Y. Or the capital R could have paired up with a small y. Likewise, the small r could have paired up with a capital Y. Or the small r could have paired up with a small y. So we have four possible gamete types. So if we look at them, here they are. And for male. Remember convention that we write the females at the top and the males on the side. It doesn't have to be like that, but it helps if you do it the same all the time for your candidates to know what's going on. So there you can see all the different gametes, each with one allele for each of the characteristics. There we have it again. And if we go and we cross, we will see that there are 16 blocks. Now, if we start with the phenotypes, this is what we get. If we look at the if we start at the phenotypes, number one, top left, capital R, capital R, capital Y, capital Y. It will be round and yellow. Round and yellow, round and yellow, round and yellow. The middle line from top right to bottom left will be totally heterozygous. Then you can look at the pattern. Dominant, homozygous, heterozygous. Recessive homozygous, recessive or dominant homozygous. There's only one chance out of 16 for a P to be wrinkled as well as green, bottom right. Now, they're not going to ask you to do this crossing. What they will ask the candidates is they will describe, as we had here, they will describe the characteristics. They will say what is dominant and wrinkled. They will describe the parents. They will say two heterozygous parents for both characteristics. And then they will start asking, what were the gametes? What were the possible genotypes of this parent or that parent, etc. So they must be able to interpret a block such as this. Here we've got the pictures of the peas, and there you can see. Let's just sit down and work step by step, block by block. And practice is the best thing. And I will send you examples. So just have a look. Sorry. Okay. In the end, though, characteristics are determined by genes as well as the environment. We know the story between nature and nurture. The flowers on the right maybe grow well because they get the right amount of sunlight and water and feeding. If not, they could have been smaller, not blooming so profusely. They could have had long etiolated stems if it was in the shade. We don't know. But genetically, they are supposed to look like that. They're hydrangeas. The little girl, it looks like a little girl on the left, could be our next Einstein. If she doesn't get the right, well, she didn't get it while still in her mother's womb, and if she doesn't get the right nutrition, the right stimulation, what will become of her? So yes, genetics and environmental factors will determine who and what we and all organisms are in the end.
Lorraine,
Yes.
if I can come in there just about this, in what extent would they use the term epigenetics now? Will it be in a sort of a question describing what it is, and the candidate must use that to talk about this? Or is it just for interest sake at the
This
moment?
is just for interest sake. I will be amazed if in future years they use the term epigenetics. It's not in any of the curriculums yet.
Okay.
It's something to know about. What Susara is referring to, epigenetics, actually the influence of the environment on your genes. In other words, not just on your phenotype, but epigenetics, certain environmental factors can literally switch on or off certain genes in your genome. So, but it's not yet part, it's for interest sake. And same with eugenics, which is where which is totally unacceptable. Okay, we use it for in animal breeding, but eugenics is actually a term used to describe where so-called non-suitable humans were either killed, as they were in the Second World War, by the Nazi camps, not just Jews, also homosexuals, intellectuals, Roma people, disabled people, and by other Western countries. People who were mentally disabled women were sterilized without their knowledge and consent. So that is a can of worms that I'm not going to open any further. You've had it glimpse, but it's not genetics. Okay, then. I think this brings us more or less to the end of our presentation as far as facts are concerned. We made good time, and think if you still have questions, ask them now. I'm going to refer you to 4 books. I looked at my bookcase and I saw She Has a Mother's Laugh. It was written by Carl Zimmer. He's a well-known evolutionary and biology journalist. And he wrote this brilliant book. It's about his little daughter and the way she loves like her mother. Did she inherit it, or was it a learned behavior? And then he continues with inheritance. And Guns, Germs and Steel by Jared Diamond. It was published quite some time ago, but it tells us how war and disease and industry shaped our world, changed and influenced our genes. And also published some time ago, but still one of my favorite books, In the Blood, God, Genes and Destiny. Also, how genetics influence the world. And I think you know Sapiens by Yuval Noah Harari. He also wrote Homo Deus, and Questions for the 21st Century. Can't remember how many questions. He's an expert on the future, and I think we should all read more and more. And then any book, we're not going into evolution now, but genetics touch on evolution. Then one of the most valuable websites that you can use is the WCED e-portal. You just type in WCED e-portal or WCED curriculum support, and you will find masses of resources, and you can contribute your own. You will find lots of resources. You will find, if you go to lessons, and that is the link that I have there, our DCAS, Mr. Jean Goliath, did the curriculums with lots of information, questions, facts that they must know, examples, problems, action words, etc., etc. Then also go to YouTube and type in telematics and look, and the topic, in this case, it will be genetics or monohybrid crossings or any of the genetic topics that you want. And there will be presentations, either by me and Colin Oppold from a long time ago, or the new presenters doing excellent work. But yes, you will find YouTube videos. Then also, I don't know, some of you might remember the Teaching Biology group, TBP, Teaching Biology workshops that we had. So go and look at TBP. They also have the Teaching Biology project. They also have videos on various topics for different grades. That, Sheila and colleagues, Rizal, Sora, brings me to the end of my presentation on the introduction to genetics. If you have specific topics that you would like to have a presentation about, please let us know. If you have more questions, Sora, are there any more questions?
No, nothing yet.
Not at this stage. And then I want to thank you for your presence. And I hope, as I've said, this was introduction to genetics. We didn't go into the more complicated issues. That, I think, is a story for another day.
Thank you very much, everybody, and enjoy your time with your learners, this beautiful subject.
Thank you, Lorraine. So, Sarah, we really appreciate it. Thank you for the time and effort that you've put in to do this webinar. And I'm sure the educators also really enjoyed it. As I said before, we have recorded and we will send the presentation.
Thank you.
And yes, we're looking forward to the next webinar. Can't wait to see that.
Yes, thank you. And thank you for your work,
Great.
Rosal and Sheila. At All Copy, I think these webinars are worth our while. And thanks for the thumbs up, everybody. Thank you.
Thank
Keep
you,
well.
everyone.
Good luck
Bye-bye.
for the rest of the
Bye-bye.
term. Bye-bye.
Thanks, Lorraine, Sarah.